Hey rice fans, welcome to another episode of Rice and Advice. I'm Dr. Jarrod Hardke, Rice Extension Agronomist with the University of Arkansas System Division of Agriculture. Here today we're going to learn a little bit about mid-season nitrogen timing on a lot of our conventional varieties and how that relates to the DD50 rice management program and how we need to relate that to optimum timing for our mid-season applications in rice, and also how to look for some of that joint movement, internode elongation, how to measure and sample for that and try to be as accurate as possible in our timing. Let's get started. So at the moment we're actually standing in one of our earliest planting dates for the DD50 Rice Management Program study that we conduct every year. So really from a yield and performance standpoint, we plant 6 of these planting dates beginning in late March and going all the way into June each year, both at Stuttgart location as well as Pine Tree. Stuttgart remains the focus for our plant sampling portion of this because that's historically where it's been conducted. So essentially we plant a wide range of new cultivars and even some that they're still dominant on a lot of acres currently to compare them not only in performance but also developmental goals that we can then put into that DD50 program to forecast what development stages we're going to reach at different timings. So essentially we go in here at just a few of those timings, really the late March, the mid-April, and then preferably the early June timing to actually plant sample for hitting that green ring or beginning internode elongation, and then half-inch internode elongation timing, and then our point at which we reach 50% heading for all of these plots. So those are that half-inch internode elongation and then 50% heading are our 2 main developmental goals within rice that we use to kind of forecast everything that's going on with the crop throughout the season. So essentially we go into here in these small plots that we've planted and we sample on a recurring basis as we near that mid-season timing around internode elongation to start picking up that movement and measuring. Very, very time intensive, a lot of labor involved in pulling those samples, splitting stems, measuring back in the lab, stuff like that. But basically what we're going to look at out here is sort of how we go about pulling those samples and measuring, and so we'll take a little bit deeper dive into that and looking how we're going to pull those. So here we have an example of one particular variety plot in the field, and essentially we're just going to reach in, remove a plant. We're going to feel around until we think we have what is mostly just one plant and its tillers. We can remove that, wash the roots off as closely as possible. And we're going to have a little bit closer look at the various stems. Typically you can pick out, here we got a couple of different main stems, but we're looking for one of those larger primary tillers. We're going to get rid of some of these smaller tillers out of the way, put some of those aside in case we need them here in a second, and make sure we don't disrupt the crown in any way. I'm going to take that, peel as many of the external leaves back as we can, and very, very carefully, obviously using something sharp, we're going to take and split that stem open, again very carefully, all the way down through the crown. And in this case we're going to need, it looks like we do have a little bit of joint movement. This is where it helps to do it in a more controlled setting. But yeah, we have just a little bit of movement. Let's have a look at one more. See what we can figure out here. Again, peeling away, trying to get as much out of the way as we can. This does look like another main stem. And we see about the same result. You can see that if you were guessing at this point, you would have about a 1/4 inch movement between those, that internode space. Again, our focus is between those top 2 nodes, those white bands, the space in between it. So probably about a 1/4 inch movement at that point. Very, very rapidly going to hit half-inch internode elongation, and we'll be able to move on this plot assuming that all plants average that level. So looking at a little bit different variety at this point, we can go in here and reach in. Again, try not to thin it out too much in any one spot. I can see one main plant there that I can pull. I'm going to wash off the roots and pull it up. That one's pretty easy to separate. See the main stem that we're looking for. Much larger plant type, much thicker stem. And we actually see in this case, and maybe just a little easier to observe, you can see just a little bit more movement here than actually what was expected. I was hoping to find a green ring there. suggesting we're about to start movement, but we do have just a slight bit of separation, internode elongation there. So that one's just getting going at this point, but again, it'll rapidly be behind the other variety that we just had a look at. So that takes a very, very brief look at actually measuring for splitting stems and looking for that internode elongation in some of our varieties that we know are hitting that mid-season timing right now. There are certainly some other varieties and hybrids out here that are a little earlier in development and a little later in development than the ones we just checked out. So it's always important to keep an eye on that. Run a DD50 program report to give you an idea of when you'll hit those timings. Should always be accurate within a couple of days either direction, but that gives you the general time you're going to have to look at. Beyond that, the other important thing to glean from those reports is that mid-season nitrogen timing, again specific for pure line varieties. What we're looking for there and what's listed on the report is a recommendation that we have been flooded for a minimum of 3 weeks since the pre-flood nitrogen was applied, and we be at least at beginning internode elongation or green ring or beyond that point. The much safer approach beyond just those minimums is to actually be about 4 weeks past when we actually established a flood on that field, and to be at about ½ inch internode elongation or even slightly beyond that. Most of the recent research, past 7-8 years, led by Dr. Rick Norman, Dr. Trent Roberts, and myself, has really shown that not getting in too big of a hurry with that mid-season nitrogen timing on varieties is actually critical to getting the most out of those applications. So we want that large pre-flood application up front, then we're going to follow it, again, a minimum of 3, but preferably more like 4 weeks after the flood has been established, and then be after green ring, probably closer to ½ inch internode elongation, again, to be absolutely certain that we are done taking up that pre-flood nitrogen. It takes, generally speaking, 21 days to take all of that up. So once we've been flooded and that nitrogen has been incorporated, it's going to take at least 21 days for the rice plant to take up the pre-flood nitrogen. Depending on conditions, get some cool spells in there, things like that, and slow some development down, it could stretch out to as much as 28 days. So our safety valve there is to make absolutely certain that the rice is done taking up pre-flood nitrogen before mid-season nitrogen is applied. If we're not done taking up pre-flood, you run the risk of making a nitrogen application into that flooded soil condition when the rice isn't ready for it, and instead much of that nitrogen will be lost before the rice plant ever actually tries to pick it up. Thanks for joining us on another episode of Rice and Advice. Hopefully you've learned just a little bit about how we go about our sampling and generating some of our recommendations for that mid-season nitrogen timing and just monitoring overall rice development throughout the season. If you have any questions, feel free to let us know. See you next time.